Welcome back to my channel Knowledge Chronicle.
Have you ever wondered how some plants thrive under the scorching sun while others struggle? The secret lies in an advanced photosynthesis mechanism called the C4 pathway, a game changer for plants in hot, dry climates. All plants perform photosynthesis, converting sunlight into energy, but not all do it the same way. The C4 pathway gives certain plants an edge over traditional C3 plants, like wheat and rice. Here's what makes C4 plants different. 1. They have a unique leaf anatomy called Kranz Anatomy. 2. They can tolerate extreme heat and high light intensity. 3. They avoid photorespiration, a wasteful process that occurs in C3 plants. 4. They produce biomass much faster, making them highly productive. Now let's break it all down and understand why the C4 pathway is so powerful. Look at the inner structure of a C4 plant's leaf. You'll see something special, a ring of bundle sheath cells surrounding the vascular bundles, like a wreath. In fact, Kranz means wreath in German. These bundle sheath cells are large, packed with chloroplasts, and have thick walls that prevent gas exchange. This setup creates a separate space for carbon dioxide fixation, making photosynthesis super efficient. Want to see it for yourself? Take a leaf from maize or sorghum, slice it thinly, and observe under a microscope. If you spot those bundle sheath cells around the veins, you've identified a C4 plant. Now let's dive into the actual process. How do these plants fix carbon so efficiently? The C4 pathway, also called the hatch and slack pathway, is a two-step process that happens in two different types of cells. Step 1. Carbon dioxide fixation in mesophyll cells. A molecule called phosphenyl pyruvate, or PEM, captures carbon dioxide with the help of an enzyme called PEP carboxylase, or pepcase. This enzyme is highly efficient and works even when carbon dioxide levels are low. The result is a 4-carbon compound called oxaloacetic acid, or OAA, which is why we call it the C4 pathway. Step 2. Transport to the bundle sheath cells. OAA is then converted into malic acid or aspartic acid, which travels to the bundle sheath cells, where the next step happens. Step 3. Carbon dioxide release for the Calvin cycle. Inside the bundle sheath cells, the 4-carbon compound breaks down, releasing carbon dioxide. This carbon dioxide is now used in the Calvin cycle, the standard sugar-making process that happens in all plants. Step 4. Recycling back to the mesophyll. The leftover 3-carbon molecule returns to the mesophyll cells, where it gets converted back into PEP, ready to capture more carbon dioxide and continue the cycle. How? Unlike C3 plants, which suffer from photorespiration, a wasteful process where rubisco mistakenly binds to oxygen instead of carbon dioxide, C4 plants completely avoid this problem by separating carbon dioxide fixation and sugar production into different cells. The result is higher efficiency, less waste, and faster growth. That's why crops like maize, sugarcane, and sorghum are so productive, even in harsh climates. Understanding the C4 pathway isn't just about plant science. It has real-world applications. Scientists are even working on genetically modifying C3 crops like rice to use the C4 mechanism, which could revolutionize global agriculture by making crops more resistant to heat and increasing food production. Now we turn it over to you. What do you think about this incredible adaptation? Let us know in the comments. And remember, your one like, share, and subscribe will help us create more valuable content. See you in the next video.